Okay, I wonder how many of you you study these questions and you try not to look at the answer and try to solve it yourself. Okay, so it says here this is diagram showing a cell in the testis of a rat. Testis refers to the reproductive organs, okay, which is in prophase 1. So this one is testing you about meiosis. Okay, so this rat has got how many chromosomes in the parent cells? You count the centromere. 1, 2, 3, 4. Okay, 4 chromosomes. So how to do this? Or what questions? Yeah, the questions here. Draw in the spaces given the possible arrangement of chromosomes. So how to start it about? Okay. Yep. So this is the answer. Okay. Possible arrangement. So this is actually one of the possible answers. Of course, there are a lot of answers for these questions because there will be genetic variations, right? The way how they arrange themselves and whether they are crossing over or not in the two legs or just one legs, and so on and so forth. Okay, so if this is the answer that you propose, okay, show it by showing prove your answer correct by showing the working step. So this is the working step. So you only need to start from, what phase is this? Metaphase 1. Metaphase 1, because only when the homologs separate from each other then it is significant. So let's look at the legs over here. By the way, this is the way where you should draw the chromosome. Okay. Number one is that usually when you explain meiosis, the minimum number of chromosomes that you use will be 4. Usually it's 4 because you want to show the homologous pair and that they are different in terms of length and also the in terms of shading. One you shade, the other one you don't shade. Okay, the color. So here, okay, this one has got color. So this is homolog to one another, longer size. Okay, longer in length. And this one is shorter and they are homolog to one another. And one you shade, the other one you don't shade. Okay, if in exam you don't have these colors, right? So one you shade, the other one you don't shade. Why? Because you want to show which is paternal and the other one is maternal. So it doesn't matter as long as you differentiate by the color. Okay, so this is how you draw. Alright, so homolog to each other must stay together because as they come near to one another, this is called synapsis, right? So they must come near side by side, sit next to each other. So how about the legs over here? Okay, do I simply say, okay, this one exchanged segment, crossing over happens at both legs? Okay, same goes to this. So how do I decide? I have to look at the question. You see? So they're touching each other at the end, both end. Okay, same goes to here. Okay, it can be this end touching and this end not touching. If this end is not touching, so you shouldn't, this one you shouldn't shade it black. So that means this whole thing, this entire whole thing must be white if they are not touching in the beginning. But then the question shows all touching, right? So that means crossing over happens at both legs. Same goes to the smaller ones. Okay, so the homolog. So you label it like this, 1, 2, 3, 4. Okay, so as you go about metaphase 1, right, how will you end up having this? So after metaphase 1 is anaphase 1, right? So if until this point you still don't know how you get the answers for this, then you have to work out your anaphase 1 and your telophase 1 for you to see it clearly. Okay, you got it? For those who understand this step in method phase 1, then I'm leveling 1, 2, 3, 4, meaning this is the first possibility. All white, one long, one short. All white, one long, one short. The second possibility, one longer, one shorter, and look at the ends, both with black tip. So this is the second possibility, and so on. The third possibility and the fourth possibility, all black, one long, one short. Okay, you got it? That's how you get the answers, right?